Tim. Let me share my screen. Thanks, Tim. Thanks to John and the folks at the AViDD Forum for invitation. So following Tim's, hello everyone. So following Tim's intro on the druggability of helicases, I'll focus here on SARS-CoV-2 helicase mostly. I see. All right. So you can see here the structure of the replication transcription complex solved by Seth Dart's lab, where you see the polymerase that is mounted onto the RNA helix and two subunits of the helicase. And here is exactly the same structure, but a simplified representation where I removed most proteins and just leave here the helicase. And you can see here really that the 5' end of the RNA is projecting directly through the core of the helicase in sp13. And so you can imagine that as the helicase walks along this strand in an ATP-dependent manner, it should be associated with the unwinding of RNA. If you map all the putative druggable sites in this structure of NSP13, the most well-defined site is actually a channel that extends from the site occupied by ATP, ADP here, all the way to the site occupied by the 5' end of RNA. This channel, especially the section highlighted in sepia here, is predicted to be druggable using sitemap, which is the most commonly accepted druggability prediction tool. And if you focus on the side chains for the helicase that are lining this channel, you'll find that these side chains are exceptionally conserved across coronaviruses. So these are these side chains lining the central channel, and most of them are actually absolutely conserved across this all 27 coronaviruses for which all proteins are reviewed at UniProt, so with a stamp of approval, if you will, from UniProt. So these are coronaviruses from humans, SARS-CoV-2, SARS, MERS, so entirely, absolutely conserved, as well from bat, bovine, mouse, etc. The only exception is this alanine here in beta coronaviruses, which is a serine in most alpha coronaviruses. Actually, if you do the same exercise, looking at the genetic stability of binding pockets, but if you do this on all putative druggable or known druggable binding pockets in the structure of SARS-CoV-2 proteins, you'll find that this central channel of NSP13 is actually the most conserved across all coronaviruses, along with the catalytic site of the polymerase. More so than the ATPase site, the ATP binding site of the helicase. And it is not only the most conserved across all coronaviruses, but it is also the most conserved across all SARS-CoV-2 samples from COVID-19 patients. So exceptionally conserved site. As Tim showed in the nice animation on his first or second slide, there is a large conformational rearrangement upon engagement of the RNA. So you can see here this subdomain here is closing up on the RNA upon engagement of the RNA. And if you flip this structure by 180 degrees, again, you can see that the RNA bound form is rather relatively closed when compared with the apo form of the enzyme. And so yes, Joe Newman with Ofer Gileadi and Frank from Delft did conduct a fragment screen by crystallography, identified many fragments bound to NSP13, and the fragment bound form that they captured were closest to the RNA bound form. So here are some of these fragments. 3 of them here are squarely occupying a site otherwise exploited by RNA. This 5th fragment here occupies a juxtaposed site, which may be actually also an interesting starting point for chemical inhibition. And so chemical inhibition of targeting this central channel of the helicase, of SARS-CoV-2 helicase, is the focus of CASH challenge number 2, where CASH stands for critical assessment of computational hit finding experiments. So this is a challenge where we are asking computational chemistry and AI experts around the world to use their methodology to come up with 100 or so molecules targeting, predicted to target this central channel. And then what we do is we procure the physical samples of these compounds, these predicted compounds, and we test them experimentally. So CASH is very much modeled after CASP. I imagine that most of you know about CASP. This is a protein structure prediction challenge that has been going on for 25 years. And that really set the stage for the technological breakthrough that was AlphaFold from DeepMind in protein structure prediction. And similarly, we hope that CASH will set the stage for a technological breakthrough in computational hit finding. So really, it is a prospective hit finding competition, as the compounds that are predicted are then ordered and tested experimentally. And then all the data is publicly released with no restriction on use. So today, when a scientist reads a paper on computational hit finding, or a pharma hears a sales pitch on computational hit finding, or a disease foundation reads a grant application on computational hit finding, it's really hard to compare these methods because they are all focused on different targets. The compounds are validated experimentally with different assays and different hands. And the goal here with CASH is to provide a unifying metric, if you will, where all the methodologies are validated experimentally with the same assays on the same target and with the same platform. So if you go to the website, cashchallenge.org, you'll see that this second challenge, which is well ongoing now, is focused on the central channel of SARS-CoV-2. So we received 41 applications, including leaders in the field from all over, mostly from academia, but also from biotechs, one pharma company, and surprisingly for me, and electronics and the internet giant as well. And out of these 41 applicants, 25 passed the double-blind peer review process. And so these are the 25 participants who are about to submit by the end of the month their 100 compounds predicted to bind to NSP13. And this is here a summary of the publication track record of these 25 participants. So you can see that collectively they published 27 papers in the past 2 years or so in JCIM, which is really the journal of choice in computational chemistry. So a strong expertise, a collective expertise in computational chemistry, also published in more general medchem papers and more high-end papers. So I think this is nicely showing that CASH is attracting a diverse team of experts to work on NSP13. At the other end of the expertise spectrum, one of the participants actually is the crowdsourcing initiative FoldIt/DrugIt, where that is challenging citizen scientists and gamers to either fold proteins or, in this case, design compounds for a target. And I think it will be really fascinating to see whether the wisdom of the crowd is outperforming or not the experts in the field. And so stay tuned for that. Finally, if you want to know more on CASH, you can have a look at the white paper. co-authored by a lot of people, including many people from pharma, Bayer, AstraZeneca, Sanofi, Novartis, Merck, Boehringer, Takeda. So this is, I think, speaking to the fact that people in industry are very interested in knowing where the wind comes from in computational hit finding. And finally, yeah, I'd like to acknowledge the hard work of colleagues and collaborators at SGC Toronto working on NSP13, at EBI, the crystal screening at Oxford, and the experimental platform at CASH, with a big shout out to Feng Ling, Sumera, Irene, who are driving the assays. And with this, I'll